Hello, and welcome back to Human Evolution. Today we want to talk about the molecular evidence for evolution, and we've talked about the anatomical evidence, we've talked about the fossil record, we've talked about all of these various things that are looking at large phenotypes, but now we want to go to the heart of the matter and look at the genes. And so to start with, we want to talk about where are we going to find this molecular evidence. Well, it's going to come in the form of DNA and proteins. And it's important to remember that all organisms have DNA, and so they're going to share common ancestors. All of the variations that you see today, as we've talked about before, are based on some mutation or other somewhere. Now, there are some very different organisms that have very similar DNA, and so they are going to be more closely related than organisms that have more different DNA, something else we've talked a little bit about. Proteins, all organisms have them. They use them to perform their daily functions. All organisms, even really different ones, are going to use some of the same proteins to do the same things, and all of them use the same amino acids to build their proteins. And one more note on DNA, they all use the same molecular alphabet, right? The same nucleotides. So when we talk about molecular evidence, we're talking about changes in nucleotides for the DNA and or changes in amino acids for protein sequences over time, okay? So your first question is this: which of the following is not true when discussing the molecular evidence for evolution? Related organisms share a greater portion of their DNA. That, I don't know why there's an S on that. Sorry about that. The hemoglobin gene is less similar between humans and dogs than between humans and chimpanzees. Only DNA can be established or can be examined for establishing evolutionary differences. Molecular evolution progresses more quickly when there is no selection for the gene, and phylogenetic trees can be established using molecular evidence. Well, we just decided that related organisms have more similar DNA, and the hemoglobin gene is going to be less similar between humans and dogs than it is between humans and chimpanzees. However, it's still going to be very much the same, or very similar, because it does the same function, and that's kind of exciting. Molecular evolution does progress more quickly when there's no selection, because remember, most of the time, changes, are not good, right? They don't do good things, and so that would be selected against most of the time. So if there's no pressure on it, the mutations don't matter, and they're going to stack up. Phylogenetic trees are something we're going to talk about in a little bit, but a phylogenetic tree is a relationship, an evolutionary relationship, and of course you would want to use molecular evidence for that. So the answer to this question is C. You can examine proteins, DNA, RNA, whatever, right? Any of these molecules can be used to establish evolutionary differences. It's not just DNA. So we've been talking about how we get new genes and new proteins and things like that. And we know that new genes arise from point mutations, substitutions, insertions, deletions, those things. We have talked about chromosomal mutations, and we'll talk about those in a minute. But basically, you can track all of these changes. And when you do track these changes, you can actually start looking at the divergence, because remember, mutations will accrue after separation. And so we can compare different DNA sequences to look for changes. So these, now it's kind of hard to tell how closely related they are. It's really better to compare more than two organisms, but you can see that there are definitely differences, and they are related, but maybe not closely related. It's kind of hard to tell, like I said, unless you are using lots, right? And so in this case, you can see here, you've got all kinds of little animals here. You have a mouse, a rat, and these are genuses, by the way, so this is several mice, rats, humans, mosquitoes, fruit flies, and then this is a bacteria. So when you look at all of these, you can see, oh, look, there are lots of places where they're very similar, but every once in a while you see, oh, look, this one is different, okay? And again, different here, right? Different here, yeah, different here. And so you start to get the idea that this guy diverged way earlier than the other ones, okay? That's much farther back, right? And then you start to look and do some comparisons, okay? If you look at this fourth position here, these are amino acids, by the way. So you have these these are all T right here. Well, that makes sense. These are all mammals, right? These guys are both insects, right? And so they would be different. And so you start doing these comparisons and you can say, okay, these guys are more closely related than these guys, blah, 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 blah. Okay, now polymorphisms, that is what we call these changes. And so here you see that you've got these differences, yeah? And sometimes it makes a difference in the protein, and this is why you can look at proteins and DNA separately. Sometimes it actually makes a difference in the protein chain if this is occurring in an actual area of DNA that codes for genes. Sometimes it does not, sometimes it does. Here, changing this A to T, you change the amino acid. Here, changing the C to A, you actually don't change the amino acid at all. Same thing here, A to T does not change the amino acid. But these changes are polymorphisms. Now, proteins, their function can change even though the overall protein doesn't. Maybe something about it changes a little bit. And here's a really good example. Lysozyme is a very common antibacterial enzyme, and it's found in most animals. It's in the cell, and it does all kinds of things. It breaks down bacterial cell walls. So, like if you are having things, actually it breaks down lots of things. So it'll break down various parts of the cell. It hangs out in the lysosomes. It'll break down bacterial walls, things like that. You also find it in tears, saliva, milk, egg whites. But in some animals, you find lysozyme in the foregut fermentation. And this protein has actually evolved some very distinctive changes that will protect it from digestion in the ruminants, right? And these are the only guys that actually have it in their foregut fermentation. That's not true, actually. Langurs and Hoatzin, I think I said that right. This is a langur, it's a primate, and then, of course, this is a bird. They also have it. They're different changes that protect it from digestion. The ruminants, they all have the same changes. That also indicates, by the way, even though they have the same function, they are different in their actual chemical structure in terms of those changes that protect it. And so these guys are all very closely related. They have the same changes. These guys are not related to each other very closely, and they're not related to these guys very closely. And so you can have a number of different things going on with proteins. Okay. Now, there are other sources. Remember we talked about you can jumble genes around, you can duplicate them, you can have more than one, you can import DNA from one area to another, you can break chromosomes, you can fuse them together, and that may create new genes, all kinds of stuff. We're only going to focus on a couple of these things, and the first thing we want to talk about is duplication. Sometimes genes just get duplicated. And so when that happens, you may have a situation where you have what's called a pseudogene. One of the genes is inactivated and the other gene still functions, but then you have this little pseudogene that will be an indicator of the fact that this duplication happened. Sometimes, right, you get the evolution of a new function. This new duplication may actually do something totally different. And then in some cases, this duplication will cause a function. Let's say that this orange gene and this purple gene work together. Perhaps it'll work on this chromosome, but not on this, wow, let's try this one more time. The purple one will work on this chromosome, but the orange one will work on this chromosome. And so you have this splitting of the functions. And eventually that may lead to look something like this, and you get overall chromosomal changes. I actually would like to talk about pseudogenes in this case. All of these things are true and very exciting and lead to chromosomal changes. But duplicated genes or pseudogenes are really, really interesting because they don't code for functional RNA or proteins, even though they may have the same sequences. They're missing whatever they need to, maybe a promoter or whatever, to actually have to be transcribed, okay? And there are lots of pseudogenes, and there may actually be as many as about 6,000 pseudogenes in the human genome alone, but one of those that has many, many replications are the hemoglobin, and different families of hemoglobin have different replications, but you can see here's one, here's one, here's one, here's one, this, 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 and this, right? And so these are all duplications, and that gives a really good indicator of the relationship and when these genes evolved into these non-functional pseudogenes. And so you can really do a lot of comparison with these pseudogenes. Now, the other thing that happens that I want to talk about is the chromosome breakage and fusion. In this case, right, when we look at the great apes, our closest living relatives are the chimpanzee, the gorilla, the bonobo, and the orangutan. And when we look at our chromosomes, Chimps, gorillas, and orangutans all have 48 chromosomes, and we have 46, right? 23 pairs. They have 24 pairs, okay? And so chromosome 1, ours is a little bit different. Theirs is very similar. Chromosome 2, right? So maybe we have a little inversion or something like that, maybe a little change, duplication, whatever. But I want to focus on both chromosomes 2 and 3. In the chimpanzee, the gorilla, and the orangutan, chromosome, what we consider 2, is actually 2 chromosomes for them. And so you have these right here, and you've got this little shortening. It turns out that we have this fused version, right? And so at some point, these 2 chromosomes fuse together to give us our chromosome number 2, right? And you can see that it matches all of this, and it matches all of that. That's a fusion, okay? Over here in chromosome 3, right, there was an inversion, right, at some point after orangutans split, and we, gorillas, humans, and chimpanzees all share that. And so these are some examples of some actual chromosomal differences that arise from these different kinds of mutations. Okay, now we can use all of these things to create phylogenies, and phylogenies are the evolutionary relationships. And so it's based on a couple of premises, and that's the idea that organisms that are similar at the molecular level should be more closely related than dissimilar organisms. Distantly related organisms would be expected to accumulate a greater number of evolutionary differences than closely related species. And so phylogenetic relationships among living things are going to be derived or inferred from these molecular similarities. Okay, so, right, we have gene trees. These are both phylogenetic trees. We have gene trees and we have species trees. Now, we are going to come back to species trees when we start talking about selection and things like that. But the species tree is going to typically be based on some sort of speciation event. A lot of times that is an actual trait. And so, like I said, we'll come back to that. But here, when you look at this, you have some sort of organism that is the outgroup, and this is true here. There's a mutation, and all these organisms, or genes in this case, will share, right? These are organisms, these are genes. All of these genes share that mutation, okay? Here, there's another mutation. Gene C doesn't have it, but A and B do. And then here, there's another mutation. B doesn't have it, but A does, okay? And so that's kind of how these things work, and they're showing this distinct relationship. In terms of the species tree, and again, we'll come back to this, maybe this is, hair, right? Let's say that this is a turtle and this is a, cat, right? So maybe this is hair. Maybe this is something else when you're talking about other mammals, and then some other trait. Okay, here's your next question. Using the molecular record to determine phylogenetic relationships is based on the assumption that nucleotide sequences do not change over time, nucleotide sequences change at a fairly constant rate over time, nucleotide sequences change randomly and erratically over time, or changes in nucleotide sequences change the function of the protein, or all mutations are harmful. Well, it's got to be based on the idea that nucleotide sequences change at a fairly constant rate of time, over time, and we talked about that when we talked about mutations. That was the molecular clock idea, and using the molecular record, we can also determine time, right, because we are using that molecular clock idea that we talked about before. Okay, now it's important to also understand that just because you have a mutation doesn't necessarily mean that there's divergence, right? Maybe, maybe not. And mutations can also occur before or after speciation. And so sometimes your gene trees and your species trees are going to be different because of this. Okay, now here is the last thing I want to talk about, and this is pandas, raccoons or bears, right? For a very long time, scientists actually didn't really know which family the giant panda belonged to. And as a matter of fact, when I was going through school, it was a raccoon and a bear, and, nobody was really sure. But it turns out that when you start looking at the DNA, you can actually figure out who the panda bear, giant panda, is most closely related to. At first, they're bears because they look like bears and they climb trees. Then they do a lot of things that raccoons do. They don't hibernate and they eat bamboo, which is what the red panda does, who is also in many ways very similar to the giant panda. So, you have to figure out which traits matter and which don't. Well, when you look at the DNA, panda bears, while being, while splitting off very early, one of the earliest bears to split off, okay, they are in fact closer related to their next bear cousin than they are to the raccoon family, okay? And so you get these guys split about 35 million years, okay? And then the raccoons actually split about 5 million years after this common ancestor. Then the panda shows up around 20 million years ago, but it's off of this branch, not this branch, okay? And then that splits, then you start seeing some of the other bears later on. And then, just as a fun fact, brown bears and polar bears are actually the youngest of the bears. So that's just an example of what you can do with this DNA sequencing. So that's it for talking about determining relationships. And if you have any questions, please make sure you ask next time in class, and I will see you next time. Thanks for listening.